Bismillahirrahmanirrahim. Class, today we will discuss the development of embryo in Ephedra. In Ephedra, embryo develops by free nuclear division like other gymnosperms. First, here the zygote nucleus divides 3 times to form 8 nuclei. As a result of these 3 divisions, 8 nuclei are produced, and these nuclei are unequal in size and scattered in the egg cytoplasm. Then these are surrounded by delicate wall to form the cells, and these cells are known as the proembryo cells, and each of these can develop into an independent embryo. So this is the special type of special cleavage polyembryony which is found in Ephedra. We can see here in this diagram that this is the zygote cell, and in this zygote cell the nucleus of this divides 3 times and 8 nuclei are produced, and these 8 nuclei, after producing, these are surrounded by the delicate walls and are converted into the cells, and these cells are known as the proembryos or the proembryonal cells, and each of these cells it develops into the embryo independently. So this is the cleavage polyembryony, in which the zygotic nucleus divides into nuclei, and each nucleus develops into an independent embryo. Now we see next how this embryonal cell are converted into the proper embryos, means how proper embryo is developed from the embryonal cells. All the proembryonal cells, they start to develop into embryos, but the lower proembryal cells, it develops more rapidly, and only one reaches maturity, mean only one develops or matures, and the others are disintegrated. Now we will discuss the development of the proembryo cells into proper embryo. The proembryonal cell, it produces a tube-like projection or outgrowth which is called as the suspensor tube. The nucleus of the proembryonal cell, it is shifted to the tip of this tube and divides, and the 2 nuclei are produced which are separated by a transverse wall from each other, and the embryo now consists of 2 cells. Number 1, the terminal embryo initial cell, and the elongating suspensor cell. The embryo initial divides by a periclinal division into an upper and a lower cell. The lower cell again divides further by longitudinal division into 2 cells, and these 3 cells divide further by repeated division to produce the embryo proper. Elongated primary suspensor, this is due to the production of this primary suspensor which is elongating, it pushes the embryo into the vegetative tissue of the female gametophyte, and the embryo cell These absorb nutrients and grow into a mature embryo. The upper end of the embryo next to suspensor cell differentiates, develops into a secondary suspensor, and the lower end of the embryo it produces two cotyledons and the shoot apex. This is the one embryo initial cell, embryonal cell, in which the suspensor tube is produced, and the nucleus of the embryonal cell, it is shifted into this tube and then divides into two nuclei. The upper one is the suspensor nucleus, which will form the primary suspensor, and lower is the embryo initial cell. And then the embryo initial cell, it divides by periclinal division to produce the two nuclei by the formation of wall. And then the lower cell, it divides again longitudinally to produce the three-celled embryo, and this embryo, which consists of three cells, these cells they divide repeatedly to produce a mass of embryo, proper embryo, and later from this mass of embryo the two cotyledons are produced from the lateral sides, and then in the center the shoot apex is produced. After fertilization, when the pro-embryos are developing, some cells at the micropylar region of the female gametophyte, these are stimulated to divide. These are divided and they form a mass of cell to form a plug which closes the pollen chamber of the ovule, means no other pollen grain can enter into this pollen chamber. In this diagram we see again the developing embryo. As we have discussed earlier, these are the pro-embryonal cells, and each of these pro-embryonal cell it develops into an independent embryo, and these are the developing pro-embryonal cells in which the suspensor tube is being developed. This is one pro-embryonal cell in which the outgrowth is formed, which is known as the suspensor tube. The nucleus enters into this tube and then divides into two nuclei, suspensor nucleus and the embryo nucleus. Then this embryo nucleus, embryo initial cell, it divides again by a periclinal division to produce the two embryonal cells or two cell stage of the embryo, and then out of these two embryonal initial cells, the lower one divides by a vertical division to produce the three celled stage of the embryo, and on the upper side of this the suspensor nucleus is present, which is elongated to form the primary suspensor, and this is the three cell stage of the embryo, in which on the upper side this suspensor nucleus is present. Then from these three cells they repeatedly divide to produce the mass of embryonal cells or embryonal mass from which the two cotyledons are produced, and in center of these cotyledons here the shoot apex is developed. So today students, we have discussed the development of the embryo, and we also discussed the diagrams to show the development of the embryo in ephedra, in which the cleavage polyembryony occurs, means that embryo zygotic cell divides to produce 8 nuclei, and each nucleus can develop into an independent embryo. And out of all these embryos which are developing, only one survives, matures, and other all the embryos, these are disintegrated. Next we will discuss the development of the structure and development of the seed which is produced from this embryo.